Hello, General Biology, and welcome back. This is screencast number 3 in the Section 2-4 screencast series of Chapter 2. Alright, and we were last talking about enzymes, and now we're actually going to see how enzyme activity happens. And you're going to get to see again my fantastic artistic abilities. I know you've been waiting for this, so here's your chance. You're going to see it. So for a chemical reaction to take place, and I'm reading right along here, the reactants must collide with enough energy so that existing bonds will be broken and new bonds will be formed. If the reactions do not have enough energy, they'll be unchanged after the collision, so they need enough energy to change. So enzymes often provide a site where the reactants can be brought together and react, and the site reduces that energy needed for the reaction to occur. So I'm going to do my picture, and then we're going to go to the picture next. So an enzyme, and I'm going to just draw this kind of as a blob. This is going to be our enzyme. And I'm going to say that this enzyme is hydrase. Okay? Or hydrolase. I spelled that wrong. Let me do the hydrolase. Okay? And let's say that I have hydrogen, 2 hydrogens, and I have an oxygen that I want to chemically react. I want them to collide, and I want to make a substance out of them. These, normally, in a chemical reaction, these would be called our reactants. But when we're talking about enzymes, these are actually referred to as our substrates. They're the same thing as reactants, we just call them substrates, because this is what we want to chemically react together. So what's going to happen is this enzyme, and I want to kind of just draw the enzyme here, is going to mold itself around these substrates. So see how it's kind of molding around? This area right here that I'm etching in, this is called the active site, and the active site is where the reaction is going to take place. When we have the enzyme and the substrates together, this whole thing is called an enzyme substrate complex. And what that is going to do is it is going to force the substrates together so that they start to join and react together. Okay? So that when finally they are reacting together, you're going to get it? A water molecule, H2O. This is going to be your product. So no energy was needed in this reaction. It just molded around, kind of like Play-Doh or silly putty, where it molds around the substrates, it forces them together, and it produces the product. What the nice thing is about enzymes is they're reversible. If I need to break this water molecule down, I can break this water molecule down with the enzyme into the substrates. Okay, so that's my example. Here's the textbook example, and you'll probably like mine a little bit better. So let's say, okay, we want this carbon dioxide, and we want the water to react because we want to get that carbonic acid, so we have CO2 plus H2O, and we're going to wind up with H2CO3 as our product. And we have to get the enzymes to work. So the enzyme is going to wrap itself around the water and the carbon dioxide. So the substrate is going to bind to that so that we wind up having the enzyme substrate complex. It's going to force the two substrates together so that the product, carbonic acid, is produced. Okay, so this is what the carbonic anhydrase looks like. Here's the active site that we were talking about, and there is the reaction that takes place with that, and we get that carbonic acid. If we needed this to go the other way around, it could go the other way around as well. So in terms of the enzyme substrate complex, the substrates bind to the site on the enzyme called the active site. The active site and the substrates are going to have complementary shapes, which means that it's going to fit like a puzzle piece in there. And as you saw when I traced it, you could see how it would fit. And the fit is so precise that the active site and substrates are often compared to a lock and key model, because you have a particular lock or a particular key that opens a particular lock. So it works kind of like a lock and key model, but it's actually called an induced fit model for the way it can mold around like that silly putty or Play-Doh that I was talking about before. It's specific like a lock and key because you have to have a specific enzyme with a specific substrate, but it's induced because it can mold around. So enzyme activity, there's a couple of things that influence that activity. So as I said, we're going to do a lab on this, and you're going to look at what can regulate enzyme activity. And there are a couple of things, 3 things, that can regulate enzyme activity. One is temperature. Temperature can change the rate of enzyme activity. So let's think about this, and I'm going to show this in class. Silly putty. You're all familiar with silly putty. It's stretchy and everything. If I were to warm that up in my hand, is the silly putty more pliable? Would it mold around something more easily if it was just a little bit warmer than room temperature? Hopefully you're saying yes, Mrs. Saunders. Okay, I can hear you through this. If I were to put that silly putty in a freezer and chill it, would I be able to mold it as easily? No, I wouldn't. So temperature is going to have the same effect. If the temperature is right, body temperature, it's going to easily mold around the substrates, and it's going to cause the reaction to happen. If it's cold, if it's too cold, it's going to slow the rate of the reaction down because it's not going to be able to mold as easily. It's going to take longer for it to mold. The same idea is if I heat that silly putty up until I melt it, is that going to be able to mold? No. So temperature can affect the rate of enzyme activity. Too hot, it's going to shut it down completely. Too cold, going to shut it down completely. But just right, it almost sounds like the 3 bears, just right, the reaction's going to happen at a steady rate. pH is another factor that will affect the regulation of enzyme activity. In your stomach, you have hydrochloric acid. Hydrochloric acid has a pH of about 1.5 on the pH scale. It's one of the strongest acids. Well, guess what? Protease, another enzyme called pepsin, and another enzyme called trypsin. There's that line again. They can't work unless the pH is acidic. So your body releases hydrochloric acid to lower the pH so that protease, pepsin, and trypsin can work. What did I tell you protease digests? Proteins, right. All 3 of these are enzymes in digesting proteins. So they need a certain pH in order to work. Another thing that can affect enzyme activity is other enzymes. And we'll see this in reactions later on in the year. There are other enzymes that can speed up the rate, affect the rate of enzyme activity. So enzymes produced by human cells generally work at a temperature of 37 degrees Celsius. That's 98.6, your regular body temperature. They work best at certain pH values, as I exampled down there. Stomach acid, the pepsin, which begins protein digestion, works better under acidic conditions. And the activities of most enzymes are regulated by molecules that carry out chemical signals within the cells, and they can actually switch the enzymes on or off. And when we talk about the cell cycle, we'll talk about switching those enzymes on and off for that cycle to happen. Okay, that's the end of the chapter, and that means that we're going to have a test on section 2.3 and section 2.4. All right, enjoy.